Today we will discuss about the abstract of a new paper where they will they are able to successfully eradicate the pancreatic tumors after the elimination of these KRAS mutations. So in the pancreatic tumor cells, in order for its survival and proliferation, uh, KRAS mutations plays an important role. So if these KRAS mutations are been eliminated, then the immune system gets activated and it can act upon the tumor cells. So this is the hypothesis that they have checked, and these are the and they are able to successfully defend their hypothesis in their paper. So let us understand what is their abstract. So in the cancer cells, the KRAS mutations are there. So because of the KRAS mutations are there, they will express this PD-L1 myeloid cells, or we can say the checkpoint inhibitor. That it will inhibit the immune system to act upon those tumor cells, and this PD-L1 plus myeloid cells will inhibit the immune cells, that is T1 cells, and immune system will not be able to act upon those cancerous cells. And however, when this KRAS has been eliminated in the cancer cells, then what will happen is that the immune system, the checkpoint inhibitors does not work, and the T cells can respond against the tumor cells, and this will cause release of the cytokines by the immune system against the tumor cells, and these tumor cells will get killed, or apoptosis of the tumor cells will occur because of the response by the immune system. So this is the zoom in image. What will happen when the KRAS mutations are turned off in the cancer cells? When the KRAS mutations are turned off, so there will be expression of this FAS, and whereas when the KRAS mutations are on, the FAS expressions will be less. This is the dead cell receptors. It is also known as dead cell receptors. This will be less, and because these KRAS mutations will act upon the promoter region of this FAS and disrupt the FAS expressions on the cell surface of the cancer cells, and because of that the CD8 plus T cells will not be able to interact with the cancer cells, and this is one of the reasons. Apart from that, another reasons is that the checkpoint inhibitor receptors such as PD-L1 plus are overexpressed on the cancer cells, so that this PD-L1 overexpressions will interact with the CD8 plus T cells, and that will trigger suppression of the immune system cells. Uh, However, when the KRAS mutations are off, so those things will not happen. That is, PD-L1 receptors overexpression will not happen on the cancer cells, and these that cell receptors will be expressed on the cancer cells, and the CD8 plus T cells interact with these FAS receptors on the cancer cells and can trigger immune response that will cause apoptosis of the cancer cells. Some of the immune response are this releasing of cytokines such as IFNG, and activating these antigen presenting cells to trigger specific antibodies against the tumor cells. So these are some of the ways by which the immune system can act against the tumor cells. And the researchers are able to see that when the KRAS mutations are eliminated in the pancreatic cancer cells, they have observed the complete eradication of the pancreatic tumor cells by the enhanced T cell response. And it is with the enhanced T cell response, it will also recruit other immune cells such as CD4 plus T cells, uh, CD8 plus T cells, and the antigen presenting cells. So this will further accelerate the immune response against these tumor cells. So these are some of the these are the basics of the abstract, which is presented in the form of graphics, that what is what is the main idea on which this paper has been described. We will discuss about this paper in our subsequent videos. However, just to have a have an appreciation about the results that we are going to discuss, the understanding of the graphical abstract, or the bigger goal of the paper, is essential. So that's all for this video. If you need to understand more about this abstract or this paper in detail, you can go through the reference that has been provided in the description. Thank you for your time.